Section 8.2, the structure of DNA. What is DNA composed of? Well, it's a polymer, so it's a molecule composed of repeating units called nucleotides. The nucleotide is the monomer, and the polymer is DNA. The three parts of a nucleotide are a phosphate, sugar, and nitrogen-containing base. The sugar is called deoxyribose sugar, and there are 4 different types of nucleotides found in DNA, which we'll get to in the next slide. Okay, these are held together with covalent bonds between the phosphate and sugar. This is a covalent bond, and covalent bonds between the sugar and the base as well. What makes one nucleotide different from another is the type of nitrogen-containing base that is attached to the sugar. Each one of these nucleotides, this is a nucleotide, has a phosphate, sugar, and a base. This base happens to be thymine. Okay, this one is a phosphate, sugar, and base. This base is adenine. Okay, so we have adenine, thymine. We also have guanine and cytosine. Those are the 4 different types of nitrogen-containing bases found in DNA. Okay, and they are just one part of the 3 parts that make up the nucleotide. Again, the phosphate, sugar, and nitrogen-containing base. The 4 nitrogen bases, as I said previously, are cytosine and thymine, which are called pyrimidines, and adenine and guanine, which are classified as purines. Notice the purines, adenine and guanine, both these guys have 2 carbon rings that make up their structure, where the pyrimidines have 1 carbon ring that makes up the structure. They still have the phosphate, sugar, and base, right? Phosphate, sugar, and base. Phosphate, sugar, base. Phosphate, sugar, base. But there are the two types of nitrogen bases, right? Purines and pyrimidines. Adenine and guanine being purines. Cytosine and thymine being pyrimidines. One of the scientists that was studying DNA structure early on was Chargaff. And he discovered that the same 4 bases, the adenine, thymine, guanine, and cytosine, are found in all organisms. Now, the amount of each of these 4 bases varies, but those 4 bases were in every organism. One of the things that Chargaff found out in his studies was the amount of adenine was equal to the amount of thymine. He also discovered that the amount of guanine was equal to the amount of cytosine. James Watson and Francis Crick were 2 scientists that had a breakthrough in the understanding of DNA structure, and they came up with the idea that DNA is a double helix. They had used findings from other scientists, Chargaff being one of them, about the amount of adenine equaling the amount of thymine and the amount of guanine equaling the amount of cytosine in DNA. And with that and other scientists, as well as their own research, came up with this 3-dimensional model of DNA called a double helix. Rosalind Franklin was another scientist that used X-ray photographs to show that DNA is in fact 2 strands that are a regular, consistent width apart. So kind of like a twisted ladder is the structure that she came up with through her research. A double helix is 2 strands of DNA molecules that wind around like a twisted ladder. The strands are complementary, meaning they fit together and are the opposite of each other. The opposites would be with these rules, Chargaff's rules of base pairing, which is adenine would be the same amount as thymine, and guanine being the same amount as cytosine. They would pair up to form the rungs, this part of the ladder. Okay, the types of bonds that connect the phosphate of one nucleotide to the sugar of another would be a covalent bond. So the phosphate from this one to the sugar of this one will be held together by a covalent bond. Same thing here. covalent bonds, remember, are the sharing of electrons. So these are going to be held together by sharing of electrons. The parts of the nucleotide that form the sides of the double helix, okay, or like the outside support part of the ladder, would be the sugar and phosphate, called the sugar-phosphate backbone. So that would be all this part here, right? Sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. Remember, the nitrogen-containing bases would be covalently bonded to the sugar, so those would be that way. And this is what makes up the sides of the double helix. The rungs of the ladder, of this twisted ladder model, right, this part here would be the nitrogen-containing bases. Okay, and they're held together with hydrogen bonds. Remember, hydrogen bonds are weaker attractions caused by polarity, a charged part of one molecule being attracted to the oppositely charged part of another. These are not full charges, these are partial charges. And that's what holds the middle part, or the rungs, of this double helix together. The base pairing rules are that adenine pairs with thymine and guanine pairs with cytosine. Adenine and thymine form 2 hydrogen bonds from adenine and thymine, and guanine and cytosine form 3 hydrogen bonds in their attraction. And these things fit together, which is what makes them pair up, is that the hydrogen bonds attract and hold these things together, and helps with DNA replicating and whatnot. These base pairing rules are very, very important, and something we'll be talking about in a later section with both DNA replication and formation of RNA.